Hello everyone, and welcome to today's discussion about the skeletal muscle, particularly the length-tension relationship. We will begin our brief lecture with an overview of the contractile unit of the skeletal muscle and how we are able to flex our muscles. Then, we will discuss the mechanics of the muscle and how they relate to the length-tension relationship. Finally, we will turn our attention to the research and application of this relationship. So, if you are ready, let's begin. Before we can discuss the relationship between length and tension in skeletal muscle, we must first understand the organization of a sarcomere and how this contractile unit allows a muscle to contract. As you can see in the picture, there are several different zones that are listed. First, we will look at the stars of the show, actin and myosin. These are the proteins that conduct the contraction movement. We will discuss their roles a little more in depth on the next slide. The A band runs the length of the thick myosin. It contains all of the myosin filaments as well as any portions of the actin filaments that overlap them. The myosin filaments are only found within this A band. The portion of the A band that the actin filaments do not reach is known as the H zone. The supporting proteins we see depicted as the orange ovals make up the M line, which extends down vertically in the A band and runs along the center of the H zone. Next, we have the I band, which is the section that contains actin filaments that do not overlap myosin. The I band will not consist of myosin. In the middle of each I band is a dense vertical line known as the Z line. The Z line separates the sarcomeres. Now that we have a better understanding of the layout of the sarcomere, let us look at how it works. Now let's dive in deeper into the process of contraction and the roles of actin and myosin. In order for a muscle to contract, myosin must be able to bind and form cross bridges with the actin filaments. However, in a relaxed state, the binding sites found on actin are blocked by a complex made of troponin and tropomyosin. Contraction cannot begin until this complex is moved out of the way. Calcium is needed to bind and move it. An action potential causes the release of calcium from the lateral sacs of the sarcoplasmic reticulum and into the cytosol of the muscles. Once in the cytosol, it binds to sites on the troponin-tropomyosin complex, altering it and allowing for the exposure of the actin binding sites. Now that the sites are exposed and ready, myosin must be charged so that it can bind to the actin. This is done with the breakdown of ATP. The myosin breaks ATP down into ADP and inorganic phosphate. Now fully charged, it binds to the actin site. When it successfully forms a cross bridge, it releases the ADP and inorganic phosphate, which causes a release of energy. This energy causes the myosin head to bend or engage in a power stroke, pulling the actin filaments horizontally towards one another. Another ATP molecule binds to the myosin head, allowing it to release from the binding site. This process is repeated with a new myosin head attached while another is being released, until the desired length of contraction is reached. In order for the muscle to relax, the binding sites must be covered again. This is where the sarcoplasmic endoplasmic reticulum calcium ATPase pump, or the SERCA pump, comes into play. This pump collects the calcium that was released into the cytosol and moves it back into the lateral sacs of the sarcoplasmic reticulum. With no calcium present, the troponin-tropomyosin complex moves back to its position over top of the actin binding sites, preventing further cross bridges with the myosin heads. Let's shift gears and discuss the mechanics of the skeletal muscle. Each muscle has an origin and an insertion point. The origin is the portion of the muscle that is attached to the more stationary part of the skeleton. If we look at the picture of the arm and focus on the biceps, we can see the point of origin is located on the scapula. The insertion point is the end of the muscle that is attached to the part of the skeleton that moves. Here we can see the insertion point is located on the radius bone of the forearm. Looking at portion B of the picture, we can see that the muscle is attached in such a way that it creates a lever to allow for the limbs to move. The biceps create tension through contraction that pulls against the weight of a load located at the hand. This tension moves the arm along the pivotal point called a fulcrum, in this example, the elbow. This placement allows for the most efficiency in moving the load against gravity. When the muscle is relaxed, this placement allows the muscle to reach its optimal length for contraction. Why is it important for the muscle to be at optimal length? Well, let's take a look at the length-tension relationship. Now that we have learned the basic components that make up and allow for the length-tension relationship, let's discuss its importance. On the previous slide, we mentioned optimal length, which can be seen as the peak in the picture located at a muscle length of 100%. This is the length at which maximal force can be produced, or in other words, the length in which the most tension can be achieved. Looking at the sarcomere marked A, we can see that there are maximal number of cross bridges and actin sites available for contraction. Let's see what happens when the muscle is stretched beyond optimal length. When the muscle is passively stretched, like at point B, at around 130%, there is a loss in the number of cross bridges that can be formed due to less of an overlapping between actin and myosin. With this, there is also a decline in the amount of tension the muscle can produce if it were to begin contracting at this length, as seen by the downward slope. As the muscle is continually stretched, there are fewer and fewer cross-bridging possibilities, until eventually, as depicted in point C, the actin and myosin no longer overlap and therefore cannot bind at all. At this point, no tension can be built because contraction cannot occur. The muscle also loses tension when the muscle is shortened, as we can see at point D. The myosin filaments are pressed against the Z-lines of the sarcomeres and can no longer continue shortening the muscle. We can also see an overlapping of the actin filaments from opposite sides. When a muscle is shortened beyond 80% of its optimal length, there is less calcium release into the cytosol. The reasons why this occurs is still unknown. Due to the skeletal structure of the body, muscles can only differ 30% from optimal length. Other measurements have been determined in a lab with muscle samples. The length-tension relationship is important for normal muscle function. When a muscle is damaged, such as tearing the tendon of a bicep, the muscle length becomes altered and the tension capabilities decrease. There is a treatment option for such an injury called tenodesis, which is a reattachment of the bicep tendon. When the length-tension relationship is not restored during this treatment, the patient can experience deformity in their bicep, as well as a cramping in the muscle and early fatigue. As discussed in an article by Tal David and Jeffrey Schildhorn, there is a development of a better form of treatment called arthroscopic suprapectoral tenodesis, which allows for more consistent results of a restored length-tension relationship. They found that by attaching the tendon at a suprapectoral location, there were less instances of deformity and cramping. In other words, the muscle behaved similarly to the way it did prior to injury. They are also looking at different anatomical locations that may exhibit the same results. And that concludes our discussion today over the length-tension relationship of skeletal muscle. The length-tension relationship is an important aspect of our skeletal muscle system and pertains to our everyday function. I hope that you found this video informative and that it supplemented your current studies and/or interests. As always, I wish you good luck in the rest of your studies and have a wonderful day.